everyone. It is Wednesday evening, September 30th, the last day of September. Hope everyone's doing well. I don't have a lot in terms of announcements today, other than that quiz 5 is now live on Canvas. And that today I am a microbiologist lab along with accompanying instructional videos on how to use your portable microscope is also under the lecture recording. I made a separate video under the lecture recording tab specifically on how to use a mobile device. But then the main instructional video for using a portable microscope is actually at the end of the biological macromolecules video. So check those out and let me know if you have any questions. Other than that, we'll go ahead and dive straight into this evening's lecture, which is going to focus on cell structure. This is found in chapter 4 of your textbook. Okay, so same schematic that we've looked at repeatedly throughout the course thus far. It's basically our guide for where we're going. We of course started out over here in ecology for unit 1. And now for unit 2, we've been building life out of its very base physical components. And so we started out talking about biochemistry, different types of bonds that are important for biological processes, ultimately justifying how important water is in the context of how life works. We spent last time going over a few examples of molecules, very big molecules that play very important roles in what life is actually made of. So the 4 types of biological macromolecules, that would be right here. And then today we're going to actually bump up to the cellular level and talk about various structures of cells. And we will just keep going on from there. So just as a reminder to what we talked about in the last lecture, because it does have a fair amount of moving pieces to it, and it's necessary for us to have a pretty thorough understanding of what we talked about last time in order for us to talk coherently about cells. And so these 4 types of biological macromolecules, sorry for my son in the background, a little bit distracting there. These 4 types of biological macromolecules are pretty important for what cells are actually made of. And so just as a quick review, carbohydrates, we're mostly concerned with functions and components. So carbohydrates are the first type of biological macromolecule we talked about, the base components of which we defined as monosaccharides, which can form these long chains called polysaccharides. And arguably one of the more important carbohydrates in terms of individual monosaccharides is the monosaccharide glucose, which forms long chains called starch. Very important for short-term energy storage. Other examples of monosaccharides include things like fructose, which can form long polysaccharide chains that called cellulose rather, which are really important for plant structure, for example. And so broadly, carbohydrates are important for energy as well as plant structure and among other things. So a pretty important type of biological macromolecule. The second type of macromolecule we talked about are lipids, which you can broadly, colloquially call the fats. And so these are things that are involved in long-term energy storage, as well as an actually really important structure for a plant organelle. We'll talk about that here shortly. They have a few broad components. One is a glycerol head, as well as a long fatty acid tail. And this will make a lot more sense when we talk about cell structure here shortly. Pretty important, but broadly involved in long-term energy storage as well as structure to a certain extent. The third type of macromolecule we talked about are proteins. Proteins, and this proteins, contrary to the colloquial understanding of proteins, have a huge array of functions relative to just simply muscle. They have lots of different functions. They can be enzymes. They're important for structure, for storing things, for transporting things. Because there are a lot of different base building blocks to proteins. We talked about amino acids, which is the base component of proteins, a total of 20 different types of amino acids. They can form these long chains, much like monosaccharides forming polysaccharides in the context of carbohydrates. Amino acids can form these long chains through peptide bonds that allow for any number of different permutations, kind of like Legos. And because of those almost infinite number of permutations, there's many different types of proteins, which leads to many different types of structures, which leads to many different types of functions. And so proteins are a very large class of biological macromolecule, very crucial to how living systems operate. And lastly, we talked about nucleic acids, which are broadly involved in energy storage. And we're going to talk a lot more about this in the third unit of the class, things like DNA and RNA, and actually a third thing called ATP, which is actually huge for energy as well. And so these are the 4 types of biological macromolecules. I would like you to be broadly familiar with the base components as well as the functions for each of these 4 categories, because it is essential that we understand these things as we begin to build cells and talk about how they function. Okay, so with that in mind, let's go ahead and promote ourselves on to the actual cell level. First of all, we have to talk a little bit about the history, because the history here is actually pretty interesting and relevant. So first of all, we'll talk about this one individual by the name of Zacharias Janssen. He was a Dutch optical maker. He actually made spectacles in the 1600s. And Zacharias Janssen is really broadly associated around the time when the first microscope was developed. Here's an actual picture of the microscope that he made. Some folks consider him to be the actual individual who invented the compound light microscope, but there is some contention there. Generally speaking, he's credited with that, but there is some controversy surrounding that. He actually was a quite colorful character. He was, as I said, he was a Dutch spectacle maker, but he kind of had a kind of a rambunctious childhood. He was in trouble a lot with the local authorities for being in street gangs and so forth. And he actually got in trouble as an adult for multiple times for making counterfeit coins. And so his credibility is a little bit suspect, but he nevertheless was quite a reputable spectacle maker who's broadly associated with being one of the people that was developed the first compound microscope. Okay. A little bit further on, about 40 years later, an English physicist and engineer by the name of Robert Hooke was in the prime of his career. And 40 years later, microscopes had developed a little bit more beyond Zacharias Janssen's time. So they, something a little bit more sophisticated that was mounted, a little bit higher resolution. 300 times was about what they were able to achieve by the 1650s. But Robert Hooke was one of the first people. He wasn't really a biologist. He was a physicist and engineer by training. He was actually a contemporary of Sir Isaac Newton. They knew each other quite well. In fact, they were a little bit opponents. They didn't like each other all that much. Anyway, they were working around the same time. This was around the time Isaac Newton was sitting under the apple tree and proposed his laws of thermodynamics for anyone who remembers those. Anyway, Robert Hooke was working with a pretty early version of a microscope and was looking actually at the bark of a tree. It may have actually been a cork from a wine bottle, which is basically the same thing. And he was looking pretty closely, at this tree bark, and he noticed that, at 300 times magnification, there were these little structures here that kind of looked like prison blocks, and so he called them cells. He didn't really know what he was looking at, but he was actually quite an avid notebooker. Another good example for why notebooking is an important scientific practice to develop. So he in his notebook noted how these cell-like structures were in cork. He didn't really know what he was looking at. This was, again, before people had a firm understanding of what the microbial world was like. So he was actually the person who coined the term cells, okay? And again, he was a contemporary of Sir Isaac Newton. They didn't like each other all that much. And in fact, after Robert Hooke died, this is kind of a side story, after Robert Hooke passed away, Sir Isaac Newton actually allegedly tried to throw away the only portrait of Hooke. They detested each other that much. And so this was actually a remake of the original portrait. And so we don't have, this may or may not be what he looked like. Again, an English physicist, okay, who coined the term cells. But this was before anyone had a firm idea about what cells were. He just coined the term. If we fast forward a couple hundred years, in the mid-1800s, these two German biologists by the name of Schleiden and Schwann, it's kind of fun to say, Schleiden and Schwann respectively. These were the two individuals, they worked quite closely together, they knew each other. They were the actual two people to actually posit what we will shortly define as cell theory. So they were the two people to formalize that, okay, cells are the basic unit of life. Schleiden, they did this within a year of each other, but they also worked closely together. Schleiden first posited that cells were the basic unit of animal life, so things like you and I, and Schwann was a botanist, so he was kind of more my kin in terms of the profession, and he was the person to actually posit that cells were the basic unit of plant life. And so within a year of each other, they posited that cells were the basic unit of life, and they broadly posited this theory of cells. Okay, so cell theory was devised by Schleiden and Schwann in the 1830s, which has a number of different components to it. First of all, its most base contention under cell theory is that all organisms, it doesn't matter what it is, if it's alive, it's composed of cells, okay? And therefore cells are the most basic unit of life. It's the most basic unit of what we can call alive. Unlike the biological macromolecules or even the organic elements that we talked about previously, those don't quite cross the threshold into what we consider to be alive, even though they form the base building blocks of what we can consider life. The macromolecules and the organic elements, SHOPs, those atoms and molecules by themselves are not considered to be alive. But when you build them together in tandem, and when different carbohydrates and lipids and proteins and nucleic acids are all kind of mosaically put together in a complex fashion, the result is the cell, which we've now crossed the threshold into what we can call a living thing. Okay, so they're the most basic unit of life. Okay, so these are the, 1 and 2 here are the more primary posits of cell theory. All living things are made of cells, and cells are the most basic unit of life. Okay. Thirdly, Schleiden and Schwann posited that cells arise from pre-existing cells. That cells don't just pop out of nowhere; they descended through time, and this was, a little bit actually before, this is actually a contemporary of Darwin. So, this was actually before evolution via natural selection was a widely accepted theory. This was all kind of happening at the same time, a period of very rapid development within the field of biology. But cell theory still had the thought to posit that cells arise from pre-existing cells. They don't just arise from nothing. And there were some actually pretty interesting experiments that were going on around this time. Louis Pasteur, who was a French microbiologist, which I'm not going to talk about here. That would just get into too much history. Did some experiments to demonstrate this. Fourthly, that hereditary information is passed on from cell to cell, much like parent to offspring. We'll talk much more about that in the next unit. All cells have the same basic chemical composition. This was actually before people firmly understood the biological macromolecules we've just gotten done talk about. So this was really much more of a theory back in those days. And lastly, energy flow occurs at the cellular level. And we'll talk about all these different things in much more detail as we progress. But this is cell theory, the most primary components of which is that all living things are made of cells, and that cells are the most basic component of life. So all life is made up of cells, as we've just discussed in the context of cell theory. Okay. So we can broadly differentiate now at this point between unicellular organisms, which are individual organisms. They're not, you remember cell theory posits that cells are the most basic component of life. And so we can differentiate between things that are unicellular, uni meaning one and cellular meaning cells, unicellular organisms that are one cell big. Okay, so this is a paramecium, for example. It's one cell wide, and yet we consider this to be an organism. Okay, it is alive. Okay, even though it's only one cell, one single cell, there are no other cells that are made up of this single organism. That is a unicellular organism. Versus multicellular organisms. And so you can have a lot of different cells that are interacting together that individually are living things by themselves, but they can't really survive by themselves, but are rather part of multicellular organisms. So this is almost getting into a community type of interaction. So multicellular organisms where different types of cells all work together for the benefit of a singular organism. So for example, red blood cells all interacting with other types of cells like white blood cells and you name it, all the different nerve cells and so forth that make up complex multicellular organisms. Okay, so all life is made up of cells. Organisms can either be unicellular, which is one cell big, or a multicellular organism, which is on the order of thousands to trillions of cells. Okay. So, from literally one to trillions, this is, quite broad. So how do we observe cells? As we've already talked about, all the way back to Zacharias Janssen and Robert Hooke and Schleiden and Schwann and other people who I didn't mention, they were all using microscopes back in the day. So microscopy is the primary way that people observe cells. Our human eyes do not have the ability, unaided, to see most cells. There are a couple of weird exceptions, which we're not really going to talk about. But for the most part, without the aid of a microscope, we do not have the ability to see cells. And so here on this scale, for example, here's what our eyes can see. We can generally see stuff relatively easily on the order of about 1 millimeter we can see. So things that are about this, a grain of sand is about as small as our eyes can see without aid. Okay, so frog eggs, for example, is just at the threshold of what we can barely make out. Okay, so as we get a little bit smaller than a millimeter, when we get down into things that are 1/100th of a millimeter, which is the micrometers, we begin to need microscopy as a discipline to actually see things. And so, light microscopes, which is literally using, light to illuminate, and different focal lenses to see, in a much higher resolution, we can begin to see things like cells. some of the largest cells around the order of a few micrometers wide, with a couple of weird exceptions. And so we can see things like, multicellular cells in a big field of view, or even single cellular organisms. We can see these with light microscopes. Okay, but even light microscopes begin to reach the end of their capacity at right around 500 micrometers or so. Okay, so they, right around the time we get to the organelle level, which we're going to talk about organelles here shortly if you don't know what that means yet. We can't make out a lot of organelles with light microscopes, because they just do not have the necessary resolution to be able to do that. At about 500 micrometers, light microscopes reach the end of their utility. But we also talked about last time another type of microscope, which are called electron microscopes, which actually do not produce, you're not actually seeing the actual image of what you're looking at. You're viewing like kind of an imprint, which is the result of an accelerated beam of electrons that are beamed really intensely onto a specimen, and then you're looking at the imprint of that. And so electron microscopes, because the electrons are so small, they can create pretty amazing imprints, very high resolution imprints that can go way down into the nanometer, so the thousandths of things, really small organisms, or parts of organisms rather. So things like viruses or DNA or individual proteins, which we're kind of looking at here. Large proteins, which are this polypeptide chain, even a little bit into single amino acids. And so electron microscopes can go, very fine resolution down to the nanometer level. And, and even, the fanciest of the fancy electron microscopes these days can just barely make out what atoms look like. And again, atoms are really on the theoretical level to a certain extent, but there are some images out there that, claim to actually be looking at atoms, which is a little bit contentious, but electron microscopes looking at very small things. One second, Harry. Okay, sorry about that, everyone. So as I was saying, electron microscopes have pretty fine resolution down to the nanometer level, and in some instances can even make out individual atoms. Okay, so broadly speaking, that's even finer resolution than we're going to be concerned with for talking about cells. We're mostly looking at things in the context of light microscopes, which generally can make out a lot of cellular features that biologists are concerned with. So there's two major categories of cells, and you may remember this from high school biology. The first category are called prokaryotic cells, okay? So prokaryotic cells are all unicellular, meaning they all are individual organisms that are one cell big, okay? So this is, without exception, versus eukaryotic cells, some of which are unicellular, so there are individual eukaryotic organisms, but for the most part, eukaryotic cells are multicellular. For example, we are eukaryotic organisms. All of our cells, with the exception of our gut microbiome, which is another whole other story, our actual bodies, our skin, our hair, our blood, all of that is eukaryotic in terms of their cellular structure, okay? So prokaryotic, which are all unicellular, and eukaryotic, which are all, well, mostly multicellular, some unicellular. Okay, so this actually harkens back to last unit. Remember when we talked about the Linnaean system of classification developed by that Belgian biologist, Carl Linnaeus? He, from the domain level all the way down to the order and class and family and genus and species. Remember that? The Linnaean system of classification. The largest tier under the Linnaean system of classification was the domain level, okay? of which there are 3 domains. Okay, so under the Linnaean system, all life falls into one of 3 domains. Okay, so these are the bacteria, the archaea, and the eukaryota. Okay, and so thankfully, the domain eukaryota gives it away. The eukaryota, so things like plants and animals and fungi, other types of pretty basic algae, like protists and things. These are all eukaryotic. Okay, whether they're single cellular like some protists are, or multicellular like most like plants or animals or most fungi. The eukaryota domain, these are all eukaryotic organisms in terms of their cell structure, versus the domain bacteria and the domain archaea. These are all single cellular prokaryotic organisms. And so I'll ask you to know that the eukaryota, the name gives it away, these are all eukaryotes, but the other 2 domains of life, the bacteria and the archaea, these are all prokaryotes, okay? So we're actually going to talk more about prokaryotes a little bit later on in the course. For now, we've just acknowledged them. Okay, prokaryotes are single cellular organisms. They are the bacteria and the archaea. Okay, and they have relative to eukaryotic organisms, they have a basic cell structure. Okay, they don't have all of the various organelles that we're going to talk about shortly. And so internally and externally, their structure is relatively simple compared to the eukaryotic cells. But that does not mean that prokaryotic cells are simple. When I took microbiology as an undergraduate, it was a whole class. It largely focused on prokaryotes. And so there are people who spend their entire lives just focusing on specific genera, individual genus of prokaryotic cells. So their physiology, their morphology, their genetics are actually quite intricate, really complex. But we're not going to go too much into detail into actual prokaryotic cell structure because there's just so much more to talk about in terms of eukaryotic cell structure, and that's what we are. So we're not going to go too much more into prokaryotic cell structure. Suffice it to say, these are things like the bacteria and the archaea, and they are all unicellular. And relative to eukaryotic cells, they're kind of simple, but not in and of themselves. They're still quite complex. So for the most part, everything here going forward, we're going to be talking about eukaryotic cells. And so everything from here on out for this lecture and the next and throughout the rest of this unit, we're talking about eukaryotic cell structure. So all of these different organelles that we're talking about, and even when we get to cell function a little bit later on, we are talking about eukaryotic cells. Okay, so over the next few slides, this is a little bit mechanistic because we're literally going to go through the different components, the different structures that are found within eukaryotic cells. We're just going to explore the different things that make up eukaryotic cells. So again, everything we're talking about is eukaryotic in nature. None of this really, well, with a few exceptions, none of this really applies to prokaryotic cells. Okay, so on the very outward exterior of a eukaryotic cell, so we're looking at this yellow structure here, there is the cell membrane. This is the first organelle that we'll talk about for eukaryotic cells. Okay, so the cell membrane, which is generally used interchangeably with the term plasma membrane. Oops, I misspelled it there. Plasma membrane. So cell membrane and plasma membrane are used interchangeably. They mean the same thing. And so this is basically the exterior of a cell. It's the outer part of the cell. Okay, so here's an actual electron micrograph. So this is actually what an electron microscope picture, an electron microscope actually took of a cell membrane. So here's a cell membrane or / plasma membrane. I may use either of those terms going forward, generally cell membrane. But here it is actually right here. Okay, so on the left here, we're actually looking on the outside of the cell, of the eukaryotic cell. And this is the organelle, the cell membrane. And on the right, we're looking at the inside of the cell. So, which is also called the cytosol. So inside of a cell, anything that's not an organelle is called the cytosol. So that's a key term for you right there. We're looking at the cytosol right here. Versus right here on the outside, we're looking at the intercellular space. So the space that's actually between individual eukaryotic cells. Okay, so intercellular space between cells and the cytosol or the cytoplasm. But that's another interchangeable term. We're looking at that's on the right side right here. So it's separated by this organelle called the cell membrane / plasma membrane. Okay, and right, this is an actual colored picture. If we were in lab, we would be actually making a wet mount yourself, where you would actually take a swab of your cheek and you would mount it on a slide and stain it, and you would actually be looking at your own cheek cells, which are eukaryotic. And so this is an actual picture of last semester, someone's cheek cells. Okay, so the cell membrane, again, is the outermost organelle right here. Okay, so all of these are individual eukaryotic cells that are part of a wider multicellular organism, obviously us. And in between each eukaryotic cell, cheek cell, is the intercellular space. Inside of each cell is the cytosol / cytoplasm. Okay, so cytosol and cytoplasm can be used interchangeably, much like cell membrane and plasma membrane. So when we're talking about the cell membrane or the plasma membrane, we are in large part talking about a pretty organized structure of lipids. Okay, so phospholipids are a type of lipid that we talked about. That's the actual structure of the cell membrane. This is why I said in the last lecture, phospholipids and their hydrophobic nature are so important. Okay, so the cell membrane is in essence a phospholipid membrane. It's composed of 2 layers of phospholipids. Okay, so here is the triglyceride component of a lipid right here, and then these long fatty acid tails. Okay, so each of these individually is a fatty acid, a phospholipid. Okay, and there are 2 layers of phospholipids here. So this top layer and this bottom layer. Okay, so another way of saying yet another term for a cell membrane / plasma membrane is the phospholipid bilayer, because it's composed of phospholipids and there's 2 layers of them. So bi means 2. So 1, 2. The cell membrane is a phospholipid bilayer. Okay, and so that, phospholipid bilayer is yet another way of saying the cell membrane. But this, just as a little side note, the cell membrane is not only composed of phospholipids. There are things like proteins and carbohydrates and even nucleic acids that are embedded into, in fact, I think I have a slide here in a second. There are things like proteins that are embedded into the cell membrane that kind of float like ships in a sea. Like these proteins are floating in a sea of phospholipids. So the cell membrane is not merely phospholipids, but for the most part, it's mostly phospholipids. So getting back to the actual chemistry of phospholipids, we talked about last time how lipids have a hydrophobic tendency. Okay, so these fatty acid tails are afraid of water, meaning they shy away from water, they don't dissolve in water. Versus the other side of the lipids are these triglyceride heads that are hydrophilic in nature. So you have this phospholipid bilayer composed of two layers of these phospholipids, the heads of which are hydrophilic, they have an affinity toward water, and the tails of which, these fatty acid tails, are hydrophobic, they are afraid of water. And so as a natural consequence of the chemical composition of phospholipids, when they're arranged in a bilayer like this, they form this solid layer of hydrophilic heads that respectively face the outside of the cell versus the inside of the cell. So the outside of the cell, the extracellular space, that's aqueous in nature. Okay, so what cells are floating around in. That's, for lack of a better term of saying it, that's a solution of water. Water is the solvent of life. And so because the aqueous environment external to cells is largely water-based, the hydrophilic heads of these phospholipids face outside. Same thing with the inside of the cell, the cytosol/cytoplasm. That's also aqueous in nature. So the other layer of the phospholipid bilayer that faces the inside of the cell, that is also faced by these triglyceride heads because they are hydrophilic. But the inside of the phospholipid bilayer, or the cell membrane, that's composed of the hydrophobic triglyceride tails. Okay, so it's because of this chemical composition of the phospholipid bilayer having respectively hydrophilic and hydrophobic components that actually forms the cell membrane. And that is actually how the cell membrane maintains its composition, maintains its integrity, because the hydrophilic heads they want to be near the water, they don't want to go away from the water. And the hydrophobic tails, they want to be away from water, they're forced towards the inside, they don't want to go toward the outside or the inside. And so that natural chemical composition is actually what maintains the structure of the phospholipid bilayer, which is hugely consequential for the structure of cells. Okay, so the phospholipid bilayer, very important for how cells are arranged. So cell membranes, getting back to the actual organelle, we're talking about an organelle in the context of cell membranes. I'll use these quotations for the next series of organelles we're going to be talking about to broadly define what these various organelles do. Okay, so cell membranes, for lack of a better term, they are the quote unquote security of the cell. They regulate what comes into the cell and they regulate how things get out of cells. So, phospholipid bilayers, that's the actual the majority composition of cell membranes. But as I said before, that's not all cell membranes are. They have, for example, these proteins that are embedded into the phospholipid bilayer. So this purple thing here is actually a protein that regulates the movement of things across. So some things that are outside the cell, like out here, that want to get inside cells, they have to pass through these proteins, or things that are on the inside of the cell that need to move outside the cell. They are all regulated by these things like proteins, other things like simple molecules that can pass directly through the phospholipids themselves. But if you want to think of it, the cell membrane itself, which is composed of the phospholipid bilayer as well as these proteins and other things, the whole thing is the plasma membrane. It is the broad outer security of cells. Okay, so not anything can just get through the cell membrane. There's an actual set of criteria, and this can get quite intricate here. But broadly, the cell membranes, the phospholipid bilayers, the plasma membrane, it's the outer security of the cell. That's the function of the cell membrane. Okay, so we've just got done talking about the first organelle in the context of eukaryotic cells, the cell membrane, aka the plasma membrane. All of this we're going to come back to a little bit later on, so we'll definitely revisit some of these terms and some of these processes. The second organelle, which we'll talk about, and I'm sure you've heard this before, is the nucleus. Okay, so the nucleus, actually, if we go back a few slides, see this dark central thing here? If you know, okay, on the outside, we're looking at the plasma membrane, of course, but this dark thing here in the center of each one of these cells, this is the nucleus. So it's an entirely different organelle that we're talking about inside of a eukaryotic cell, the nucleus right here. So here's kind of an artist's rendition right here in this lighter purple, at least, of what the nucleus looks like. Okay, so the nucleus is the quote unquote manager of the cell. Okay, oh, I guess I could have just forwarded here. This is the same picture. So here are the plural nuclei. So we're looking at a bunch of nuclei here, but an individual nucleus right here. Okay, the nucleus is the manager of the cell. It's actually where the DNA, the genetic information of the cell resides. Okay, so it's where the DNA is packaged in various forms, and so it's where the big boss sits in the center of the cell that gives the direction for how proteins are made and what those proteins do and all of the other functions that are going on inside the cell, and even what the cell, how the cell behaves relative to the whole organism. The actual, headquarters, the corporate headquarters of the cell is located right here in the nucleus. It's where the DNA resides, okay? And we're going to talk about the function of the nucleus in much more detail in Unit 3. But the nucleus is the manager of the cell. It's where the DNA resides, okay? The third type of organelle we'll talk about is called a ribosome, okay? So the ribosomes, and ribosomes, it's not just, in the case of the cell membrane, and in the case of the nucleus, there is one of each, okay? There's one cell membrane. There's one nucleus. But for ribosomes, there are many individual ribosomes per cell, okay? They can exist floating in the cytosol, kind of, freewheeling, but they can also exist embedded in a structure here. We're a different organelle type. They can actually exist embedded here. So here's these little blue balls are ribosomes. Okay, so there are many ribosomes in an individual cell. Okay, so ribosomes, okay, is another organelle type of which there are many per cell. Ribosomes are protein factories, quote unquote factories. And so let's go back to actually this picture right here of the nucleus. The nucleus is the manager of the cell. It's where the DNA resides. Okay, it's where all the information for how cellular processes are carried out. It's where it actually lives. Okay, but then at some point, the orders from the manager have to leave the corporate office and they have to go to the different satellite companies from the base. And so ribosomes are one of the different satellite organelles that take their direction from the manager, the nucleus. And so what the actual ribosomes do, they're actually the factories of proteins. And so they actually take the information that comes from the nucleus, and we'll talk about this process in much more detail in the next unit. So we're just kind of giving a broad introduction here. But the ribosomes take the information from the nucleus and actually produce proteins. And proteins, as we talked about, are composed of amino acids, and there's many different types of amino acids and many, many different types of proteins. And so the ribosomes are the actual factories that produce proteins. Okay, so it takes that information from the nucleus and it comes to the ribosome, and the ribosome actually manufactures proteins, and the proteins are what do the vast majority of all cellular work. Okay, so this is the site of the actual synthesis of proteins. So a very important type of organelle, of which there are many in an individual cell. Okay, and this fourth organelle type we'll talk about is actually a bunch of different organelles. If this were a majors level class, you would be required to memorize and understand in detail all of these different structures individually. But I just think that's a little bit unwieldy for the purposes of Biology 11. So maybe in high school you had to memorize them, but I just think it's a little bit too much busy work, and I just don't see how it's it's just too much detail. I used to teach all of them individually, but we're not going to do that. I haven't been doing that for a while. So I'm going to broadly define these series of different organelles as the endomembrane system. Okay, so there's a number of different organelles here that are involved. Things like the Golgi apparatus or the endoplasmic reticulum. They're kind of fun to say. Lysosomes. Okay, they're all in and of themselves vesicles. Okay, they're all in and of themselves different organelles. Okay, so but collectively, I'm just grouping them all together into this thing called the endomembrane system. And so this actually does a bunch of different stuff. Okay, it's broadly involved in taking finished products and packaging them up and distributing them to other parts of the cell, or broadly involved in taking trash and collecting it from site A and moving it to the, getting rid of it, dumping it outside the cell. So, it's kind of a logistics center, like an Amazon warehouse. Okay, so I define it as the Uber of the cell. The endomembrane system, which we're kind of looking at right here, the reds and the greens, this is the Uber of the cell. It's broadly involved in taking things and kind of repackaging them into different forms, or transporting them from one part of the cell to another, or even getting rid of things entirely, transporting waste out of the cell. It's the Uber of the cell, the Amazon warehouse of the cell. So it moves things all over the place. Cells are complex things. Okay, so they have a variety of different products that need to go from place to place, and so the mail system that moves things from one part of the cell to the next, that is the endomembrane system, which again is composed of a bunch of different organelle types: the Golgi apparatus, the endoplasmic reticulum, vesicles, lysosomes, and so forth. I'll just ask you to be aware of the endomembrane system and what it does broadly, which is transporting things to various places and repackaging things. Another type of organelle is the mitochondria. Okay, so this is actually really important. If you think of cells as cities, okay, the cell membrane is the outer wall, like a medieval city. The outer wall that regulates how things are able to come in and go out. The nucleus is the central keep. The ribosomes are kind of the smithy. Okay, like I'm kind of using medieval examples. So where things are kind of made. The smith. The endomembrane system are kind of like the couriers. Okay. And finally, the mitochondria, kind of more in terms of a modern city, the actual power plants of the cell. Okay, so this is where actual energy is burned that is used to fuel cellular processes. And again, we'll talk about the actual fuel that is burned a little bit later on, but the mitochondria is the actual site. And here's an electron picture of what a mitochondria actually looks like. There are multiple mitochondria per cell, just like in the case of ribosomes, but here's an artist's rendition. In and of themselves, these are actually really intricate organelles. There's a lot of complexity. If we were talking about cellular respiration, which we're not right now, we're going to do virology instead, we would largely be talking about mitochondria. but we're not talking about them right now. Suffice it to say, mitochondria are the power plants of cells. It's where the actual energy is burned, that, broadly involved in taking glucose and breaking it down into this thing called ATP, which we'll talk about in much more detail later. Mitochondria are the power plants of cells. It's the motor. It's how things are kind of broken down and used to create energy for the cell. Okay, and so these are actually really complex organelles. Look at they have an external membrane, the outer membrane. There actually has another membrane inside the membrane, called the cristae, and the matrix is the inside of the inner membrane. There's a lot of morphological and anatomical complexity to a mitochondria, as you can see here again in the actual electron microscope image, an external membrane, and then the internal cristae you can see quite distinctly, and then inside the cristae is the matrix. This is a little bit too much detail, but the reason why I'm mentioning is that there's an actual theory that poses that mitochondria, they're actually kind of cells in and of themselves, even though in the context of a eukaryotic cell, they are organelles. The reason why is that mitochondria and even chloroplasts, which we'll talk about here shortly for plants, mitochondria and chloroplasts, they actually have their own genetic information. They don't have nuclei, but they have DNA and RNA. They have ribosomes that manufacture their own proteins. And so they're kind of cells within cells, okay? We're talking about them here, we're defining them as organelles in a eukaryotic cell, but they're kind of cells within cells. So some biologists have posited that way back in evolutionary history, okay, we're going back billions of years, mitochondria were actually primitive cells. And at some point along the evolutionary history, they were engulfed by other eukaryotic organisms, and they became dependent on and subservient to the wider eukaryotic cell. And so at one point, it's posited, it's a theory that mitochondria, which are these orange structures here, and the same thing in the case of these chloroplasts, another organelle type in plants. At one point in time, billions of years ago, it's thought that mitochondria and chloroplasts existed as independent primitive cells, but at some point they became engulfed by other cells, and today they no longer function as independent cells. So this is the endosymbiotic theory of mitochondria. Endo meaning inside, and symbiotic meaning it's kind of a mutualism, or interdependent. And so the mitochondria no longer exist within by themselves, but they perform a very crucial role as organelles inside other cells, which is the factories of cells, or the powerhouses of cells rather, that are broadly involved in making energy. But at one point in time, it's posited under the endosymbiotic theory that they existed as independent cells, which is pretty fascinating if you think about it. This gets kind of into the more the tier of evolutionary biology, but I thought I'd mention it because it's pretty interesting and relevant. Okay, so moving on with our exploration of the different organelles in eukaryotic cells. The next, we're getting near the end here, so bear with me, deep breath. The next organelle that we'll talk about is called a cytoskeleton, and this actually has a bunch of different types to it in itself. Things like microtubules, which you'll hear this term again. Take note of microtubules, a very important thing that we'll talk about in this unit. Remember microtubules, microfilaments, okay? These are all the cytoskeleton, so I'm kind of grouping things together here as well. But the cytoskeleton broadly, if you consider it as an organelle type within eukaryotic cells, these are the cell scaffolding, okay? So they're the actual inside the cell. It's kind of shown here by the microfilaments here in purple. They're the actual structures in cell that kind of hold the cell together. They're like the internal framework that kind of helps the cells maintain their integrity, but they also do other functions. In the case of microtubules, which you will hear again, I'm stressing this, hint, hint, very important. Microtubules, which are involved in a number of tasks. The cytoskeleton, the scaffolding of cells, pretty important. You'll hear this again. And lastly, I believe this is the last category, the seventh category of organelle we'll talk about, the cilia and the flagella. Okay, the flagella, here's one shown here in the case of a human sperm cell, and the cilia, which are multiple things here on the on the surface of a lung cell. Okay, these are the cell propellers. Okay, for lack of a better term, the quote-unquote cell propellers. They're involved mostly on the outside of cells, but also sometimes in the insert of cells that kind of usher things along. They help cells move. Okay, so in the case of human sperm cells, this is a flagella that kind of uses a corkscrew motion to help the actual cell move from place to place. So a lot of cells, in terms of how they actually move around in the intercellular environment, they make use of things like flagella, versus cilia, which are more multiple in structure. They can exist outside of cells as well as inside cells. They are kind of involved. They kind of have waving, fan-like motions, which are also involved in kind of ushering things along. So in your lungs, for example. Another example is our throats. Our throats are actually coated. Our throat cells on the outside are actually coated in a fine layer of cilia. And so whenever you have a cold and you got all that nasty phlegm that goes into the back of your throat, that when your throat is all caught up, stuck up with all that nasty stuff, have you ever noticed how your throat has a natural tendency to kind of cough that stuff back up? It's the cilia that are involved in kind of pushing that mucus back up your throat. The cilia are the actual organelles that are involved in kind of pushing things, ushering things back along. So the cilia and the flagella are the cell propellers. They kind of push stuff. They push entire cells, or they push stuff that are outside of cells, even sometimes inside cells. Okay, so the cilia and the flagella. Okay, so these different, we've up until now, we've been talking about these different organelles that are endemic to eukaryotic organisms, eukaryotic cells. Well, with a few exceptions, some of them also exist in prokaryotes. We're not going there, but we've been talking about all types of eukaryotic cells up until now. But to make it a little bit more specific, let's differentiate between animal and plant cells. Animals and plants are all in the domain eukaryota. Okay, so they're all eukaryotes. Animals and plants, they all have cell membranes. They all have cell membranes. They all have nuclei. They all have mitochondria, which are these little balls here. They all have an endomembrane system, so the Golgi apparatus, the lysosomes, the endoplasmic reticulum. They all have a cytoskeleton. They all have mitochondria. All of these different organelles, which we've just finished exploring, they belong to both plants and animal cells, which are, again, all eukaryotes. But animal cells have some more organelles that are not part of animal cells. So plant cells are even more complex than animal cells because they have everything that animal cells have, but then they have a few more structures that do not belong in animal cells. There are a couple of other differences as well. Plant cells tend to be a little bit bigger, okay, so on the order of 10 to 100 micrometers in length, whereas animal cells tend to be a little bit smaller, on the order of 10 to 30 micrometers. Typically, animal cells are round or irregular in shape. Go back to that slide of the endothelial cheek cells back on the nucleus slide. Those are animal cells. They kind of have an amorphous, irregular shape to them. Some of them are kind of rounder. Okay, so those are all animal cells. typically round or irregular, versus plant cells, in addition to the additional organelles we'll talk about here shortly, they're typically rectangular or cubic in shape. And so they're much more kind of geometric in shape. And we'll talk about why that is here shortly. And so plant cells have everything that animal cells have, but they also have some more organelles, which animal cells do not have. Okay, so the first type of organelle, which occur in plant cells, but not in animal cells, are cell walls. Okay, so cell walls are not cell membranes. I know it's a little bit confusing because they kind of sound like the same thing, but they're not. Cell membranes or plasma membranes are phospholipid bilayers. They're composed of lipids. They occur in plants and in animals, but cell walls are entirely different, and they only occur in plant cells. So cell walls, they're not composed of phospholipids. They're actually composed of carbohydrates. So it's a long chain of carbohydrates, which it's a polysaccharide. Actually, cellulose is the polysaccharide type that makes up cell walls. And so here's an actual image of individual plant cells. Every one of these is an individual cell, and we can actually see the plant cell walls here on the outside. These dividing barriers are the cell walls. Plant cells tend to be a lot more locked and rigid, and as I said, cubic, in box shaped in their structure, as opposed to animal cells. If you look at an animal cell under a microscope, they kind of more are floating around. Look at those endothelial cheek cells in the other slide. This is more typical of a plant. If you look at a plant under a microscope, they're much more kind of locked in, in large part because of the cell walls, which are carbohydrates. They're distinct from cell membranes. Okay, and so this is what actually gives cells plant cells their physical structure, their rigidity. Because plant cells, they don't have skeletons, or plants don't have skeletons, of course. And so if you ever kind of come to a woody plant and you knock on the wood, and this thing doesn't have a skeleton, what makes it so rigid? It's, if you get down to the cellular level, it's the cell membrane, which is composed of cellulose, which is a carbohydrate, a polysaccharide. Okay, so cell walls, plants only. Another type of organelle which only occur in plants is chloroplasts. And maybe you've heard of chloroplasts before. These are broadly involved in photosynthesis. We'll talk more about photosynthesis in the last unit of the course, but broadly, as I'm sure you know, broadly involved in taking the energy from the sun and converting that energy into chemical potential energy with water and carbon dioxide, and ultimately making things like glucose, which is a sugar. So plants can make their own food. And that glucose can be eaten by other things like heterotrophs, animals, and that's broken down in our mitochondria. We're kind of getting into ecology there. But chloroplasts are the actual organelles which the process of photosynthesis takes place. Here's an artist's rendition here. Here's an electron microscope picture of what a chloroplast looks like. Okay, so just like mitochondria, chloroplasts are thought to have at one point been primitive cells in and of themselves under that endosymbiotic theory. They have their own genetic information, they have their own ribosomes. They're really intricate, as you can tell by their internal structure. They are thought to have at one point been their own cells, but now they are subservient to plant cells. They are the site of photosynthesis. And lastly, the central vacuole, which is this broad structure here. This is where plants, they store water and other secondary metabolites and sugars and things. It's basically where plants store their, let's broadly go ahead and define as that's where plants store their water. So this is actually what gives plants their structure. If you think about plants as a balloon, an individual plant cell as a balloon, when a balloon is fully blown up and tight, that's because it's, a plant cell is full of water. Its vacuole is full of water. But let's say you haven't watered your house plant for a while, your garden plants for a while. What tends to happen? The plant begins to wilt and to droop. That's because the reserve of water in the individual plant cells is depleted. Okay, so it's a loss of pressure inside the plant cells, which is actually called turgor. So the pressure that kind of keeps plant cells nice and happy and plump and full of water, that's actually when the vacuoles are nice and full of water. When the plants don't have as much water as they need, the vacuoles shrink, and that's when plants actually begin to lose their internal turgor pressure and they begin to wilt. So vacuoles are another organelle type which are only found in plant cells. And with that, that is it for the different organelles of eukaryotic cells. I know that's a lot, so fear not. We are going to explore these different organelle types in greater detail throughout this unit and the next. And you'll have some lab activities that have to do with them as well. So with that said, I'm going to end it tonight because I'm already over an hour. But just as a reminder, this week you are working on the Today I Am a Microbiologist lab, which has two components, doing notebooking on 5 different organisms using your portable microscopes, as well as using the record function on your computer or the MScopes app to record yourself pondering, I observe, I wonder, this reminds me of, for one of those 5 organisms. So I'd like you to upload a notebook scan as well as a video recording. There's 2 separate portals on Canvas that are live now for this week's lab, which is the Today I'm a Microbiologist Part 1. And then you have the Quiz 5, which is now live. As of this lecture, you have all the material that you need to take that quiz. So I'll leave you alone for now, but please reach out if you have any questions. I'm available via email. You can send me a video email or a written email. We can meet on Zoom. So let me know if you have any questions. Except for that, our next moving piece, you got that lab and that quiz due on Monday, and I will meet you on next Monday, which is going to be in October now. I think October 5th is next Monday at 8:30 p.m. We'll be meeting live again on Zoom for a lecture on cell function. So, all right, everyone. I hope you're doing well. Please do not hesitate to reach out if you have any questions or concerns. All right, good night.